have to run labs on the floor due to that's where the outlet being, and they got some fabulous results. Now, for all the stress this year has caused, to be able to give the students something, even if it's not as much as what we would do in person, really has been worth it. We got to micropipette, make solutions, run gels, isolate DNA, do an ELISA, and now we're going to be sending DNA off to be sequenced. Yes, there's going to be mishaps, students dropping, spilling reagents, or family members moving their reagents in the fridge or freezer so that the student can't find them. But it quickly became really evident to me that lab days are my students' favorite days and are actually fun. Real quotes from students. I observed that more students, even ones who are more frequently absent, showed up for labs, and we got to do lots of fun things like having scientists virtually come do labs live with the students while they're doing the labs at home, or students doing labs with siblings. Something I would never get to do in person is, the sibling watching over and, wanting to be involved in the lab my students were doing and inspiring students from a younger age that they can do this. However, I will admit that I do look forward to being back in person so students can get more proper lab training and gain more independence in carrying out labs. However, I really am extremely grateful to Mass Bioed and MiniOne because they made something that in September I couldn't even imagine happening so real for me and my students, and really have given us some great moments despite the pandemic this year. So I'm really grateful to them, and thank you so much for letting me speak and share my experience.
Hi, I'm Rachel Tenenbaum, and I'm at La Jolla High School. Go Vikings. And the school is about 1/4 bussed in and socioeconomically disadvantaged. And we get kids from all over, despite the name La Jolla High. It is a public school in San Diego Unified. And I teach juniors and seniors in a biomedical pathway. That's a 4-year program. And this year I had 46 students in the junior class, which is the one that I'm going to talk about with respect to sending materials home. So next slide. So here you see the suitcases that were generously provided by biotech, and I can't thank them enough for everything that they did. They actually prepared all the materials, aliquoted all the materials for me, so I didn't have some of the logistical issues that my colleagues did. So that part was great. And I wheeled these out onto the street, and for a couple afternoons, I had students come and pick them up, and I checked them off and wrote down the serial numbers. And then when they brought them back, which took a little bit longer, I checked them off again and made sure that everything came back. So 46 students doing the PTC taste test, because if we'd have been in person, we'd have tested our own samples and done the PCR and been able to see what their genotypes were. But since we weren't in person, we had to do it a little bit differently. So they were able to do this at home. And I broke it up into 3 days. The first day, I talked about PTC and how that was discovered and the genetics behind it, and how you make a standard curve, because it was really important to me that the students know how to use a standard curve and interpret unknowns from a standard curve. And then the second day, we went over practice pipetting and gel loading. And so I used a dock camera to show my students what I was doing in real time and for them to follow along. And I taped everything, also recorded everything, and told my students, if you want to just follow what I'm doing, and then once class is over, do it on your own, you will have a recorded version of what we did. I did feel after the fact that it would have been nice to have had little snippets of, this is how you load a gel, and this is how you melt the gel, and all those things, because I did have some technical difficulties with my dock cam off and on, and kids not being able to see it. So I went through that on day 2. And then day 3 was to actually do the activity where they ran the gels and did some post-lab questions. Next slide. So on the 4th day, after we had done the lab, and what you see here is a picture of a student at home doing their sample gel loading so that they could practice before they actually loaded the gel. But one of the really great things was that MBTech packed an extra gel for the kids I had a kid who had their gel cup explode in the microwave, so they didn't have a gel. So there was another little package of extra gel so they could just pour it in the cup. I did also have the kid who didn't have a microwave, which is something that now I would ask ahead of time because again, you want to be able to know ahead of time so you can work around those issues when it's right in the present. It's like, okay, does your neighbor have a microwave, or your friend across the street? So then I put the kids into breakout rooms because we were still completely distant at that point. And I gave each group a topic that I wanted them to focus on from the lab. So the first group was focused on the actual genetics of PTC. And so they were going to present the Punnett squares and all the different combinations that you could have based on the inheritance of PTC. The second group was going to predict what they thought would happen in the gel and why they thought the gel would look that way based on what they knew about the characters in the scenario that they were doing. Group 3 worked on the pedigree chart. So they would take the information that they knew and the information that they would get from the lab and put them on a pedigree chart for the other students to see. Group 4 used just a standard graph to try to calculate those 3 unknown bands in the PTC gels. And group 5 was using a logarithmic scale to calculate those unknown bands. And so they could see that one was going to give them really good results because they knew what those unknown numbers were supposed to be. Whereas the standard graph wasn't going to give them very good data. And group 6 was to talk about how taste receptors work and how that cell signaling happens, because what we were looking at was basically the ability to taste this, this particular paper with the PTC. And then the last group was going to take the results and explain them. And I invited the head of our CTE program to come, and Richard came from MBTech, and I think Erica also as well, to see what the students had gotten out of this experience. So it was all about, can they take what we did as a lab and really associate it with what we'd been talking about all year long. Next slide. So you can see on the right are a couple of the pictures from those student presentations. You can see the actual gel that the student did at home and all the bands and all the lanes that are labeled. So they had, Jillian was their test subject, and she couldn't taste, but everybody else could. And so they were looking at, well, why couldn't she taste if that's recessive? But yet her parents could taste and her brothers could taste. And so that was the scenario that they were looking at. And then on the bottom right, you can see one of the graphs that they came up with where they can show the actual band sizes that they calculated based on the standards that they had on that first lane in the gel. And some of the things that the students basically said was, it was really great to actually be able to do this at home, that they really weren't expecting that this was something they could do while they were, distant, and they really appreciated it, that they were using the tools that we always talk about using, the pipettes and the gel boxes. Some of them felt like they needed a little more help. They found it difficult to do something like this at home without the teacher like right there to answer their questions, but that they still got a lot out of the experience, even though they struggled a little bit with it. So it was challenging, but it was still a good experience for them. And I think that's my last slide.
Hi, I'm Dr. Barbara Huncosa, and I'm the faculty and department chair for biotechnology at Maricosta College, and we're located in North San Diego County. Next slide. So I'll give you a little bit of perspective on our programs and just kind of moving into the community college space for at-home skills. So our students can be interested in kind of any area of biotechnology, from research where we're coming up with new products like a COVID vaccine, all the way up through clinical testing and then just large scale production of products. And we have a variety of certificates and degrees that students can earn. And I'm going to talk a little bit today about the kind of foundational lab skills class that's part of our certificate of proficiency that is the base for all of the other certificates and degrees in our program. And one of the things I'll point out is that students interested more on the production side, there are various stackable certificates that lead to an associate degree and then to a Bachelor of Science in Biomanufacturing. We are one of 15 community colleges in California that were authorized to offer a baccalaureate degree program. And we chose to focus ours on biomanufacturing because we are in a big biotech hub down here in San Diego. Next slide. So the goal for us was really to continue to offer hands-on courses during the COVID pandemic. So a little bit about our student body. The average age of our program students is about 28 years old. We often have students who are older, kind of coming back, retraining, changing careers. Many of them are working full-time and are parents or taking care of their own parents or other relatives. So our students often have a lot of personal commitments and work commitments outside of school. Many of them are economically disadvantaged. And you can see that a fifth of them are housing insecure or food insecure based on their own reported insecurities. So essentially what we're saying is that our students are looking to move into the workforce, right? They're looking to complete certificates, degrees in order to gain employment. And for that reason, we have a lot of hands-on courses in our program to help students gain the skills they need to move on into the workforce. So stopping our education during the COVID pandemic really isn't an option for our students. And we have seen a huge uptick in hiring during the pandemic in response to COVID. So we wanted to make sure that our students are continue to be well positioned for these careers. Next slide. So our solution was to create really robust at-home kits for our foundational laboratory classes. I teach the first one, BTech 110, Basic Techniques in Biotechnology. So this course covers everything from solution prep to assays, to culturing cells, purifying proteins. So a lot of the basic skills that someone would need in the industry. We then also created at-home kits for our PCR course using the technology from biotech, as well as our upstream bioprocessing, where students learn how to culture bacteria and yeast. So you can see here kind of the staging in our lab. Our lab has become just a land of boxes and bags so that we can prep these kits and then students pick them up outside in our parking lot. And then I've also created a lot of videos using GoPro cameras that I'll wear either on my head or I'll have next to my hands on the bench top so that students can get kind of a point of view video of what they'll be doing and what supplies have been provided to them. Next slide. So in terms of my class in specific, which is the basic lab techniques class, students come to pick up and drop off their kits. So they come to campus 4 different times to pick up reagents and equipment. And then the faculty have created many videos to support their at-home learning. Because our students have so many commitments, we don't have live lab sessions. The students work at their own pace. So oftentimes they're doing experiments at night or on weekends. So it's important as faculty that we are responsive to them on email when they run into issues. And then students upload their notebook documentation and images of their results. So in order to help students get through the experiments, we've created lots of videos. And down here, there's an example of a short clip from one of our videos. Here we're using the biotech Mini 1 gel electrophoresis system. And I basically just made this video with my cell phone. I put it on a box above where I was working so that students could see me loading the gel. And then I have kind of edited in there shots of me from afar so they can see how I'm holding the micropipette. I also edit in different little lecture pieces to explain to them the theory of what's happening. And then on the right-hand side there, you can see just an image from a student notebook where they've loaded a gel. And then this particular student took pictures every 20 minutes. So they had a time series in their notebook. But that way, as faculty, we can see how things are progressing, and we can also check on their data analysis. Next slide. Oh, here's the video. So I'm showing them how you would load a gel. And then the video now cuts to kind of me from the front where you can see how I'm holding the micropipette so that students kind of get a sense for what a good technique might be to keep their hands steady. And then it kind of cuts to a more theoretical lecture portion. So we have lots of different videos to help support students with lots of images as well. Next slide. So students end up completing about 11 different labs at home, and this is anywhere from making salt solutions at different concentrations and then checking their conductivity, to monitoring the growth of E. coli over 12 hours in their house, to isolating DNA from bacteria and then running it on the gel electrophoresis system. So lots of different activities, very similar to what they would do in the laboratory. We just had to make a few adjustments based on the fact that they're working from home. But we were really able to recreate the majority of the course in their homes. So students are working individually, but we have watched that they often congregate on social media like Discord in order to share images and share data. And the one thing that has been nice is that for students, they can have as much hands-on time as is possible or as they desire. They've had the micropipetters at home since the beginning of the semester, so they've had many weeks to practice whenever they have a minute, so they can really master that skill. Of course, the downside is not everyone's home environment is conducive to learning, so there have been lots of distractions, and kind of being that support for them by email or Zoom is really important, particularly on weekends. Thank you.
All right, welcome everyone. So hello there. So I'm Victor. Currently I'm a postdoc at UCI, but I'm teaching at some local community colleges, such as Santiago Canyon College at the moment. I will be teaching at Irvine Valley College and Fulton College as well next semester. So I'm basically an adjunct professor around the local Orange County community colleges. And one particular class program that I'm teaching in is basically the Orange County Biotechnology program. You can go to the next slide. So particularly, this is kind of like a summary of all the classes that you can take in order to get a certification program. For what makes my program kind of unique as well is that we're focused also on dual enrollments. Basically means these courses are offered for high school students. So before they graduate, they could already have the ability to, sorry, phone. They could already have the ability to kind of take all these classes and get to certification by the time they graduate. So this is a huge advantage. Right now, we're building a class, particularly Bio 190 and 190L, particularly 190L for online. And it's been quite a challenge. And I'll tell you why that is. You go next slide. So for 190L, of course, when you transition everything to online versus in person, there's many differences. So I'm currently right now I'm teaching an in-person lab as well as teaching an online lab, and I could see the following difference. For one, in-person lab, they could we could take a picture of their notebooks and they have all the information there. On online, they could either write a notebook for themselves or they could just type it up, which is flexible, which I give my student instruction. For the in-person class right now, I'm teaching at a community college, while the online version, I'm teaching at a local high school. So there's going to be two different kind of concepts going on there. For the local high school, what we usually do is that we schedule one time and we package as much materials as possible for them to bring home with them. So we'll deliver these packages to their local school, and the high school student will come into their school to pick up those package, having a sign-in sheet, making sure they gather all the supplies, and also a sign-out sheet, or once they return by the end of the semester. Okay, you go to the next slide. So some of the differences between the online version and in-person version. So I'm going to have my syllabus, you can see on the left side, part of it. I'm going to go over one section at a time. The first portion of it is just laboratory safety and notebook and conversion and measurement and using a micropipette. They're all very similar, exactly the same concept. The students, online students, could learn what the in-person students can do. But the next part, when we start going to the aseptic technique, using agar plates or microscopes, spectrophotometer, it becomes completely different. So for example, when we're growing bacteria in an agar, on an agar plate, in person, they have an incubator. This is very convenient. But online, they don't have an incubator. I mean, they can use an oven, but we don't want to grow bacteria in an oven. So an alternative, what we can use is something like a desktop heater or your laptop. It creates a lot of heat, could culture bacteria. Another thing here, the online class does have a microscope, which is pretty expensive, a light microscope. But the virtual, for the online one, we'll use a virtual microscope that kind of have the same flow, the same idea, but it's not really something that's physical. So that's a huge difference there that we have. In person, in regards to when they're trying to learn dilutions and learning about absorbance, the online class, they have a pH meter. They could use that machine in addition to their pH paper. They also have a spectrophotometer to read the absorbance. However, we don't have a pH meter or spectrophotometer that online class could bring home. And so the best they can do is either do a dilution test, like a serial dilution, to determine the concentration based on visual observation, or just use a litmus, the pH paper, and that's the best they can do. And so that's a huge difference in learning different techniques. But overall, they're able to learn the concepts. And lastly, the same thing here, they have also learned about pH and enzymes. For the in-person class, we're using like amylase, which is like an enzyme found in your saliva, breaking down starch. We can't really have the student store amylase in the refrigerator or the freezer, and so we have to come up with something that's more sustainable. So that will be protease, and they're basically found everywhere. And a good form of protease would be like meat tenderizer salt, right? They have a lot of protease there too, as well. So we use that as an alternative there to do the enzyme test. You have next slide. And final differences I'm going to mention here between online versus in person is some experimental differences would be, well, before I go there, this year's is the Amgen gel electrophoresis. When they're running the gel electrophoresis, it works the same way, the online and in person. Very great, actually. But when it comes to further on, when it comes to more like restriction digest, transformation, inoculation, induction, or cell lysate and column chromatography, the only problem we have there is the students, there's no experiment that we have for this online students. And that's the big concern that I have there. We're still working on developing methods, an alternative for students to take home or at least learn the technique, such as column chromatography. But at the moment, we don't have any experiment for that. And so instead, we're gonna have the in-person class. The students there, they'll record what they're doing and post it on YouTube so that the online class have the opportunity to see what's going on and what they've missed. Okay, and and same thing as the last few weeks of class, they don't have the ability to do PCR. And so only the in-person has the ability to do PCR using the machine. And so overall, I really think the online classes the last half of the class, they're missing a lot of experience what the in-person class would have. And so that's my main concern about that. You could go next slide. One thing I do have the students to do is a group activity. So the question is it possible to have online class have the same experience for group activities as the in-person class? And I say, yeah, it is possible, so long you have a certain, depending on what group project you have. And this is some examples of some group projects here, and the students able to create their own group project, work together over Zoom. I've also noticed that for the in-person class, they also use a Zoom to work together, right? So it's very similar to what the online class will have. So they don't just work together in class, they also work together over Zoom and discuss among each other. So this kind of activity, the group activity, is still the same and is still possible even with an online class. You have next slide. All right, so overall, this is my last slide. So as a conclusion, a few things I noticed between the online class and the in-person class. Also keep in mind my online class are high school students, while my in-person class are community college students. I've noticed that the online class seem to be more shy in comparison and less collaborative or interaction, so they don't interact with each other. And so you're required to kind of come in and give them more of a clear guidance and pay attention to them. So in other words, you have to be jumping around breakout rooms constantly, right, to make sure they're talking, making sure they have the topic they need to talk about. Another one as well is that I feel that they lack the full training experience. For one, the last half semester, they're missing a lot of actual in-person training, like actual experiment they could be doing. But another one is as well is that it's very difficult to notice and correct their mistake, because sometimes they don't even know they're making a mistake, or sometimes they're too shy and they don't tell you. So that's one thing I feel like they're lacking as well. So you can't really be there and really watch them carefully, especially if their webcam is not pointing at whatever they're doing all the time. But overall, the experience that the high school students have over the online is pretty positive. Here's some of the quotes that they give me. Is one of them is like, I found it interesting. I'm very happy I'm doing this. This lab is interesting. I never seen anything before. I think this lab was very informative on how to use instrument. I might be a pipette god. I don't know. So this is just very positive feedback so far, in my opinion. So this is great. But again, overall, in regards to online versus in person, online definitely have a lot of room to improve in comparison to in person. And that's just kind of my opinion there. But yeah, that's all I have for the speech.
Thank you all so much. Really appreciate your insight and your expertise. And we have a few questions from the attendees, so please feel free to hop in as needed. The first question is, how much of your prep time has converted to video production now? Do you get any help with editing?
So I don't get help with editing, and I'm really basic. What I ended up doing was mostly videotaping what I was doing on Zoom, so that the kids who were missing out were just getting it that way. So really rudimentary, because yeah, I didn't have the tools nor the time to really do that kind of stuff. So for me, it was not too bad.
I would say a lot of my prep time, particularly in the fall, the first time we ran the class this way, was in just creating videos. Our lab staff took some videos for us, and then I did a bunch. We have 2 GoPros in our department that we use, and then I edited all the videos. So I became very familiar with Camtasia pretty quickly to create them.
I'd also say that YouTube is a valuable resource here as well. If you really need to, you can make the video, but there are a lot of great videos that I have used from there that show various techniques. Also, a lot of times made by the companies who make the kits, if you're using a kit.
Also, I have a really quick little video that if you let me share my screen, I can show you here really quick. So this is something that the biotech, you can actually put your cell phone. They have a platform, and so you can put your cell phone on there and do the time lapse on your iPhone. And so this was a really good thing to have, so that if kids weren't able to get their experiment to work, this was a way to show them what it actually looked like in a time lapse way. So that was pretty cool. And as you could see, you could see the banding pattern really nicely there. So that was a good thing.
Great, thank you all so much. Another question here. A lot of your examples are for small groups of students. Any advice for those of us who have over 100 students in our programs, please?
So, you know, I had the 46 kids all in one, they were in one group. I currently have a class of 45 right now. That's one class of 45. And one of the things that I found that worked now that I'm simulcasting is I had one student who was doing the lab and another or two more in a group with them on in a breakout room, and they could work together to get the lab done, even though only one of them had all the lab materials to do it. And then they kind of shared with each other what the results were. So that's one way you can kind of spread out the materials when you have larger groups.
Great, and what, for the larger populations, 200 or so, what additional obstacles do you foresee, please?
I foresee, I guess on my end, I think the most obstacles for working with a larger group is going to be how much materials you need and really how to deliver the kits, right? So if you're going to do labs over and over again, you can't be like me driving around the city, right, to all those students. That's not going to work. You really have to, schedule a pickup day and have multiple labs planned in advance, which really is a lot more planning about having, a lot more materials sorted for different labs in advance. So that's one way to be able to do multiple labs, or, you may not be able to do as many if you really can't have the students easily come get them multiple times a day. So it depends really how far your students live from your school. My students, again, live all over Boston and even some north and south of the city that commute to my school. So if I had more than the students I had, the methods I use, I would not foresee working, and I probably would have had to change that.
At the community college, where they've been serving larger coursework like microbiology, they've broken students up into groups. So let's say 30 students at a time are doing experiments. That does disconnect, the lab experience from the lecture a bit, but it might make it more workable where if you have a small group of equipment, you can loan it out to one group of students for a couple weeks, and then they bring it back, and then the next group of students picks it up. So you could do something like that.
The other thing I would suggest is that you could have them do different experiments so that you're not sharing all those materials. It would also require a little bit more spent on the logistics of it, but you could say, well, this group is going to do, this experiment, this group will do this experiment, and they'll watch each other's and just kind of break it up that way.
Great, thank you. We just have a short time left. Two quick questions, and then I think we can wrap up. Did you say the special iPhone stand is provided by the manufacturer?
Yeah, so I don't know if you guys can show a picture, maybe, there you go.
Great. And then one other question, love the lab grouping idea. Would that help hybrid blended classroom environments as well, please?
Yeah, so that's what I did is I'm in that hybrid learning environment right now where I'm simulcasting. And what I decided I was going to try was to actually have them all be on Zoom and put them in breakout rooms where the kids that were in class had the materials and the kids at home had the instructions. There was a virtual lab that they all did as well. So they had so they would get the background knowledge. But the idea was that the kids who were at home could read or be the observers and the ones telling them, okay, now you have to do this. And now it's time to put it in the incubator and take care of reading through the protocol while the student in class was actually doing the lab.
Great, thank you. We are just a minute over. I want to thank all of you again. Thank you to our wonderful panel. We really appreciate it. It was fantastic. Thank you to all of you for attending. Please feel free to stick around in an hour or so. The networking session begins, and also please feel free to rate this session. Thank you all again. We really appreciate it. Have a wonderful rest of your day and week. Thanks.
Thank you.
Bye.
Bye.
Thank you, Brendan.
Thank you. Take care.
Thank you, Barbara.